Dr. Berlin Nelson is a soybean pathologist who works primarily on fungal and umiceti pathogens of soybean, especially root rot organisms and the interactions of these pathogens on crops. He's been at NDSU since 1979. So you've worked with Ted Helms extensively, I'm assuming.
We sure have, yeah. Okay, can I share my screen here?
Yeah, absolutely.
I'm going to start off with a little bit different here. I want to give you a little history here of diseases in North Dakota. I think it's pertinent to the discussion. And when I first went to soybean fields, it was in 1980, the summer of 1980. And I want to bring you back 40 years ago, and you'll just see this graph here. Now, this is bushels harvested, which is just simply mimics how many acres there were. In 1980, there were about 210,000 acres of soybeans, and you had to look for a soybean field back in those days. You didn't walk across the block and see a soybean field. And I want you to just notice that at about 93 or so, how it really took off and just skyrocketed. This is data from 2017. There were 7.1 million acres of soybeans in 2017. Okay, so I want to show you this here. I think all of you have probably seen this. This is the 2017 bushels, showing all the bushels of soybean produced in North Dakota. And you can see it's all over the place now. In many counties, they didn't even record the data. But what's important to remember from a historical standpoint is that in the 1970s and the 1980s, all the soybeans were congregated down here in the southeast corner of the state. Richland and Cass counties, that's where most of the soybeans were produced. And if you were to go back to 1980 and you looked at a similar map, it would look like this one right here. There were maybe 100 and some thousand acres in each one of those counties, and that's all. So when we talk about the history of diseases, this is where it starts, right down here. And that's where growers started to get concerned about diseases. And this is a little sketch of what we saw in terms of diseases. First, Sclerotinia stem rot was the first disease that was on the radar of growers and scientists back in the 1970s. And by the time I came in at the end of the 70s, I was actually hired to work on Sclerotinia stem rot, not on soybean. It was actually on sunflower. But we, of course, had an extremely wide host range, so we wound up working on everything. But into the 1980s is when we started seeing Phytophthora Root Rot, and it became a concern. Then seedling diseases started to pop up in a complex, we think, from fusariums and rhizoctonias. Into the 2000s, we started to pick up some stem diseases, started to see brown stem rot. We started to see a little charcoal rot in field. We started to see some virus diseases. We've never seen a serious case of viruses yet, but maybe they're out there. 2003, as Dr. Yan pointed out, soybean cyst nematode was found. And then in 2018, we found sudden death syndrome. And as a matter of fact, this past year, we found sudden death syndrome all the way from the border of South Dakota all the way to the border with Canada. So these diseases just move in. And in 2020, I think that Dr. Markell probably has found Cercospora leaf blight and maybe purple seed stain. And so what's next? I'm sure there's something else that's going to pop up. Okay, so let's just talk about Phytophthora root rot. And again, one of the first soil-borne diseases that was of great concern to growers, and caused by Phytophthora soy. And it's important to remember, this is an oomycete. This is a member of a different kingdom. This is not a true fungus, even though it looks like a fungus, acts like a fungus. It is an oomycete. As a matter of fact, this group is what we call the water moles. And you'll see the importance of that when we talk about the conditions that are associated with this. And very shortly, just very quickly, I want to give you just a little history of this, the actual disease cycle. This organism overwinters in soils as oospores. These are resting structures. They will germinate. They will form a sporangium. That sporangium will produce flagellated zoospores. Those zoospores are attracted to that plant by several isoflavones that are given off by the plant, exudates from the plant. So they swim right to it. And I emphasize swim. Soil moisture plays a huge role in the development of this disease and the biology of this organism. And I might add, there are a lot of factors that are involved in the germination of these oospores. But one thing we know is this fungus, this oomycete, is pretty darn smart. They don't all germinate at the same time. Some of those won't germinate for several years. Anyway, those zoospores will insist on the plant, that young plant, germinating plant, or on the roots, and it will penetrate. It will begin to destroy the tissue systems, and it will begin to produce more oospores in the tissue. And that involves a sexual process where we go through meiosis. So in terms of the development of variation in that fungus, the sexual process and of course mutation can play a very important role. Those oospores overwinter, they just are in the tissue, they go back to soil, and that cycle continues again. Okay, there's some pictures here of what those oospores look like. You'll see this word oogonium. That's the structure within which those oospores form. That oogonium becomes a wall around it. And when you see them in the tissue, they look a little bit like a tire. I'll show you another picture here in a moment. And look like this right here, look like this tire in a tissue. This is actually a host tissue here. You can see the cell wall right there and see these big oospores. They're quite large, about 45 microns. So they're pretty big, good size. This is a Zosparangium. And that Zosparangium, you can actually watch the cytoplasm cleave out the zoospores. Those zoospores are biflagellate, and they pop out of there, that little ostiole right there, and they will go swimming like crazy over onto a plant. Okay. So what are some of the characteristics of this disease? Number one, it's soil-borne. You can't eliminate it from the soil. And it's hard to get to those things in the soil. You know, they're in the soil. Trying to get something to them is a lot different than trying to get to something on a leaf. These oospores persist for years. There's really no way to get rid of them. Even in the longest of crop rotations, you're not going to really make a huge change, I don't think. Flooded conditions favor disease. That's why the Red River Valley is traditionally the home of this fungus. Heavy clay soils and poor drainage. And, you know, it's guys like Hans Kandel that are making a big dent in there with all that tiling that they're doing out there. But, and the other thing to remember is that these plants are susceptible throughout their entire growth cycle. From the time you put that seed in the ground until the time, even when they're adult plants, if it is susceptible to Phytophthora, it can be infected and actually killed. Most of our damage, historically, most of our damage has been in the young plant stage when we have those heavy rains, flooded conditions, high soil moisture at the time of seeding and at the time of seed germination in that first month or so after planting. And this can destroy, it has the ability to destroy entire fields. And it has happened here. And of course, we have the other characteristic that gives this a real fit, and that is we have numerous pathotypes of this pathogen. And I'll talk about that. You'll notice here, it causes both a pre-emergence and a post-emergence damping off. You can see the pre-emergence up here in the upper left-hand corner. These are slides from Ohio where they really did a lot of work on this and have a true expertise in the history of this. You can see the post-germinate, the pre-emergence damping off and post-emergence damping off. Here's some more examples of it down here. Some more examples taken from right out here in Fargo of the post-emergence damping off and pre-emergence damping off. And then I want to point out to you, this is a slide from Ohio. Notice this area right here. Notice the lack of plants. And in this one, they put in furrow application of metalaxyl. And a metalaxyl is one of the very few chemicals that was described one of the very first ones that came out that would actually control an oomycete. General fungal, true fungicides would not control the oomycete. But I just want to show you, how much damage it can actually do, even in a small area like that. There's probably 90% of those plants didn't get even get out of the ground because they had a very high inoculum density. Okay. Now, this is a field in North Dakota that has Phytophthora root rot, but I must tell you, whenever we get flooded conditions in a field, the question always arises, well, the soybeans don't do good. Many of those cultivars, if you give them a week in wet water, I mean, their feet wet, they don't like it. They start to go downhill. And I know that the breeders will tell you that. But we also have other fungi out here. We've got about 7 different Pythium species that will attack those roots. And of course, under stressful conditions, you also got Fusarium, you got Rhizoctonia, and even a few other critters out there. So sometimes it's really difficult to know what is the primary cause of a situation like this. But we do know that in some of these fields, we can pick out a lot of Phytophthora at the very early stages. Once they get to this level of damage, then you can't find those pathogens because they're invaded by all the secondary organisms. And we also have the case of adult plant root rot occurring with Phytophthora. And that's generally maybe a little later in the season when we have the high soil moisture, we get germination, we get zoospore production, it attacks those roots. It may not kill the plant, but it'll weaken the plant tremendously. You get a hot day and that plant needs water and it just doesn't have the root system. And of course you get this kind of wilting. We also get generally this brown discoloration of the basal portion of the plant. And we get this Phytophthora root rot out there, okay? So that occurs too, and we can have that later in the season if we have the right conditions. So how do we manage this disease? Well, first, the most important is the use, has been traditionally, the use of resistant cultivars, and we'll talk about that. Also, this is primarily because of single gene resistance. However, we have some cultivars that have what is called field tolerance, or another term that we like to use is partial resistance. And that partial resistance works across the different pathotypes. In other words, it's not race specific. Field drainage, that's why guys like Hans Kandel are so important, because they show the growers how to reduce that level of moisture in the field, and that can have a tremendous effect on reducing the amount of damage from Phytophthora, just lowering the amount of moisture that those plants are sitting in. Chemical seed treatments can protect the young plants, but they will not protect the older plants. So seed treatments do have a role in this whole thing. And again, I want to emphasize that crop rotation really is not useful in terms of trying to reduce this particular pathogen as it is with other important pathogens. Okay, I want to just show you what we mean by field tolerance or partial resistance, because I'm not going to dwell on this, but I think it's important to know that this does exist. It is a very useful trait, and the pathologists believe that this is useful if you combine it with other methods of, for example, tiling, for example, seed treatments. Maybe a cultivar that has a high level of partial resistance, and it generally works pretty good under moderate to low disease pressure, not high disease pressure in general, okay? And I might add that as far as we know out here, we have not seen any high level of resistance to metalaxyl, but it hasn't been looked at for some time. And this is an example of what we mean by partial resistance. The upper variety right here, notice how brown the roots are, how short they are, because most of them have been killed off. And so this is a highly susceptible cultivar. But look at this one, under the same conditions. Notice the large root systems. They do have some rotten roots in there, and some of those roots have been pruned off. But you can actually get a pretty good plant develop from that, especially if they don't, if they're in good growing conditions, they don't have a lot of other stress, and actually get some yield out of that. And so that's what we talk about when we're talking about
tolerance or partial resistance. And there's a lot of confusion in those two words, but it is a useful management technique if it's used properly. Okay, so let's talk about host resistance for the management of Phytophthora root rot. Now that's really what the rest of this talk is about. RPS genes refer to resistance to Phytophthora soyae, the oomycete pathogen, okay? So there are at least 20 RPS genes that have been identified. Many of them have been well studied. Some of these have been cloned, and these have a very unique kind of immune type of response, and I'll show you what that is. And these genes are effective at the time of planting, and they can actually work throughout the life cycle of the plant. Some of these have been mapped. A number of the important ones like 1K and 1C are on chromosome 3. And there's been a lot of work done with all this, and I'm not going to try to go through it, nor do I know all the current research. However, few of these R genes have been used in the development of commercial soybean varieties that you find from state to state. Really, there's very few. And the two most common are 1C and 1K. There are some others. There has been some stacking of genes, but I think that if you just look at all the varieties around the central portion of the United States in the soybean production area, you'll find 1C and 1K are extremely common, even under conditions where there's plenty of isolates out there that will attack those two. One of the first genes that was deployed was 1A, and it very quickly became ineffective because of the development of pathotypes that would attack it. When I came here in 1980, there were cultivars that had 1A, and they very quickly really were ineffective. Okay. So let's talk about the virulence diversity of the pathogen and how it has changed over the past 3 decades. And I want to give credit to Dr. Wei Yan. He helped me with part of this study, and we published a paper just as sort of an overlook as to what happened over these past several decades. And it has some of the same data that I'll show you here. So how has this changed? Okay, first of all, we used to talk about races, and I think you all know what a race is. Now we talk about pathotypes and virulence phenotype. And a virulence phenotype is nothing more than a list of those RPS genes that pathogen, that isolate of the pathogen, can defeat, okay? So we describe these pathotypes based generally, not exclusively, but based generally on their reaction on 8 different isolines that have these 8 different RPS genes. So race 4, for example, has the pathotype 1A, 1C, and 7. Those are the 3 resistance genes in soybean that it can defeat. It can kill those plants. Okay, everybody clear on that? Okay, so let me show you what this resistance look like. When we want to test a plant, all we do is we grow the oomycete up in a little dish on some agar, V8 agar medium, likes V8, likes its nutrients, I guess it likes vegetables, huh? And we grow it up for about 7 to 8, 9 days, and then we mash it up and actually run it through a couple of syringes to get it nicely mashed up. And then we inject a little bit of that mashed up mycelium into the stem of the soybean plant. We just take the needle, push it all the way through the plant, and as we bring it back, we force a little bit out, and it just goes inside there. And within about 3, 4, 5 days, that's the result of a susceptible soybean plant. And this is a resistant soybean plant. Now, this one happened to have RPS 6. Okay, and that was one that Ted put early into his program, and it's turned out it was a very wise idea. And I'm not sure exactly why. I think it happened to be just that, he wanted to cross something, and that particular one had an RPS 6, and it turns out it was a very good gene. This one had RPS 1C. Well, RPS 1C now and I'll show you the data, is not a very good gene anymore, because an awful lot of isolates out there, pathotypes, can kill it. But that's what we talk about when we do that test of, you know, what gene is in the host, and is this host susceptible, is that cultivar susceptible, etc. Okay. So let's turn back the clock to about 1993, when we did our first look at what the pathotypes were. And at that time, we were talking about races out in the soybean fields. Now, I want to preface this by saying at that particular time, we were isolating from plants. Okay. Later on, we went and we baited it from soil. So there's a difference between the way we got these isolates, and that may have affected our results a little bit. But I think in general, the story is going to be the same. So you'll notice here in 1993 that when we tested all these pathotypes that we had, they all turned out to be race 3. Now, race 3, if I can move this over here, has a virulence phenotype of 1A and 7. So any cultivar that would have 1A or a 7, none of them had 7, but a number had 1A, would be susceptible to race 3. We did find race 4, race 1, race 9, and just a few of them. Okay, so that was the picture in the early 1990s. Okay, now let's jump ahead 10 years. We did a very large study in 2000, between 2002 and 2004, 10 years later. Now remember, the actual increase in acres of soybean dramatically increased during that time. A dramatic increase, probably from, I don't know, 700 or 800,000 back in that, to well over 1 million at that time. Okay, and much more than that. It's even more than 1 million. I forget what the number is. But now take a look. Race 4 was dominant in the soybean fields at that particular time. Now, we baited this from the soil. Okay, we have a technique where we can bait out using a susceptible soybean, and then we isolate from that soybean, and we found race 4 was dominant. Race 3 is still around, but now look at all the other virulence, all these different pathotypes, and I'll show them to you, that we find out in soybean fields. Again, keeping in mind that during that time, we had a very heavy production of soybeans, and we had an increase in the total number of acres. Okay, so lots of new pathotypes. And I want to point out to you, if you look at this map here, look at the solid circles here. The solid circles represent the soil samples that were positive for at least one or more isolate of the oomycete. Okay, and that's generally where we would find it at that time. And it's still about the same. It's still primarily in the Red River Valley, on the edges of the valley, where we have the longest history of soybean production and a real increase in inoculum in those areas. That's important to remember. Okay, so what I want you to do, these are the virulence phenotypes that we found in those years in the early 2000s. I want to point out to you this. Notice these, and notice that they have a 1K in there. Okay, so those are virulence phenotypes. Those are pathotypes that can attack the 1K gene. And if we look at that number, 19 of 157, so 12% of that population that we found could defeat the 1K gene. Okay, well, let's take a RPS6, one that Ted uses in his programs. Only 6 of 157, so only about 4% could defeat the RPS6 gene. So if we just look at those two numbers, you'd have to say, wow, RPS6 is a lot better gene than RPS1K in general out in those fields, right? But not very many cultivars actually use RPS6. It was primarily the ones from NDSU and a few others. Let's take a look at this in terms of the bar graph here, just to emphasize this. If we look at the number of isolates that could attack the 1K gene, we see it was about 12%. The attack the 6 gene, about 4%. But now look at this one here. That's the RPS, that 1C, almost 60% of those isolates that we took out of the soil in those years can attack the 1C gene.
So that really tells us that that 1C gene really was losing its effectiveness out there. Okay, and even today, there are a lot of cultivars that still have 1C in their background. Okay, so in 2015, we did something a little differently, and this is what Dr. Yan did. We decided that what we would do is we would do a small survey primarily in these southeast corner where we knew that we would have our heaviest inoculum load, but we would only look at RPS1C, 1K, 3A, and 6 because those were the genes that we would find in cultivars being sold in this area. And we had 61 isolates from 33 fields out of 81 sampled. Now, what I want you to do is to concentrate. Notice right here, this is the data from 2002, 2004, and that was the percent, the frequency of isolates that would attack the 1K gene. Notice that's about 12%. Now jump to 2015, 10 years, a little over 10 years, 11 years later, and look at that number. It's up to 40% of what we took out of those three or four counties down there. Now we're seeing a tremendous increase in the isolates that can attack the 1K gene. Okay, not too many for RPS3A or RPS6. Still pretty mild, pretty low numbers down here. Now let's take a look at the number of isolates that can attack two of those genes. Okay, if we look back at 2002, 2004, there were about 5% of those isolates that could attack the RPS1C gene and the RPS1K gene. Okay, so they had 1C and 1K in their pathotype name. Okay, now let's jump to 2015, and what do we see? We see almost 30% of those isolates out there can attack 1C and 1K. So we're seeing this really dramatic shift in the pathogen population, keeping in mind what's happening with the soybean varieties out there. They're using a lot of 1C, they're using a lot of 1K, and the pathogen is pretty well adapting to what's out there in those fields. Okay, so that's the short story of what's happened over the last 30 some, almost 40 years of soybean production here. And so I guess the question is, can we keep up with changes in the pathogen population? Will single gene resistance continue to be valuable, or will gene stacking be necessary? The data suggests that gene stacking would be a good next step. But what about partial resistance? And then, of course, the question will win, who will win? Will P. soya be the victor, or will the NDS scientists prevail? And that's it. Thank you. I'd be happy to answer any questions if there are. And I'm voting on the NDSU scientists for sure.
Thanks, Berlin. You got me very excited about Phytophthora. That was a really fun presentation. So I have a question while people are still coming up with theirs. So I have heard this, I've heard the idea that when a plant is healthy, it is less susceptible to pathogens. But since this is a soil-borne pathogen, what about other things that could be in the soil that could be proactive for the plant and then naturally make that plant resistant to the pathogen besides resistance? So are there any certain nutrients or other microbes that could outcompete?
I think it would be difficult under the, this flooded conditions that promote disease. I don't know if mycorrhizae really will have much role in it. I suspect they would have some, but honestly, I really don't have a clue as to what that could be. Anything that would favor the plant over the fungus, honestly, I don't know. I really don't. No.
Okay, this is from Aaron Lorenz. You see many 3A plus 1C stacks. Does the 1C add anything to the 3A?
Well, it can. I mean, not all fields have pathotypes that can attack 1C. I mean, we still have pathotypes out there that, that don't attack 1C. But, given the very large number that we pull out of soil, I would suspect that 1C is not adding a lot, adding some. So if you could stack something else in there, that certainly would be helpful, I would think. Yeah. Sure.
Okay, and I have one more question. Do you think that there's any interaction between SCN and Phytophthora?
I don't know. I don't think anybody, I don't know of a publication has really looked closely at that. Um, I think, I would think that under the, especially when we're talking about the seedling stage, because that's where a lot of our damage, if we get those flooded conditions at the seedling stage, I would expect that the nematodes, first of all, aren't going to be very active under those conditions, under those flooded conditions. Certainly, at least in our work, and when we looked before Guping came, when we looked at some fields that had very high soil moisture, the nematode activity was really, really reduced. So I would think that under the flooded conditions that are very conducive to Phytophthora root rot, that we would, the SCN would be, activity would be minimal and not interfere with that. But I don't have the data to support that.
You should be careful. You're going to start encouraging farmers to not tile their fields then until they can control their SDN populations. No, I'm just kidding.
Thank you.
Okay, Berlin, thank you so much for an excellent presentation.